So chapter 26, it's about the reproductive system. So we saw similarities about the female and male reproductive system, which includes that both systems have gonads, which are the primary reproductive organs. Gonads will produce hormones like estrogens, progesterone in the case of females, testosterone in the case of males, and then they produce gametes. Gametes are the sex cells, sperm cells and egg cells. So gametes, they have half of the number of chromosomes than diploid cells, which are the rest of the cells in our body. So egg cells and sperm cells have 23 chromosomes instead of 46. And then these chromosomes, they are derived from our paternal side and our maternal side. So we have paternal chromosomes and maternal chromosomes. And we have, when in our diploid cells, which are also known as 2N, N refers to sets. So we have 2 sets. So we have in our diploid cells, which are the rest of the cells in our body, 2 sets of chromosomes. One set coming from our parental side, the other one from the maternal side, and they pair up when the cells are about to divide, and this is called homologous chromosomes because they carry the same information as the other ones. And then within these chromosomes, we have segments of nucleotides, DNA bases that are called genes. And we have 2 versions of a gene, which is called an allele. Everything that we see externally is called a phenotype. And the sequences of nucleotides that has this particular order will be the genotype. And then, although you have the genes that codes from the same trait that you can see, for instance, eye color, height, hair color, skin color, we have different versions. And this is called the alleles. So then we said before that sometimes our parents may have a different eye color than you have. But why is this? Well, because their gametes, they have information from the parents from them. And then the parents from the parents. So we create diversity with these alleles. And then we have a change of information, genetic information, to create diversity in the gametes during the process of meiosis. Meiosis is making gametes, sperm cells and egg cells. And then when an egg cell is fertilized by a sperm cell, you form a new structure that is called the zygote. And the zygote will undergo different changes that ultimately will lead to the formation of an embryo and then a fetus. And then the zygote will have 46 chromosomes because it receives 23 chromosomes from the sperm cell and 23 from the egg cell. And then in meiosis, we reduce the number of chromosomes in the cells by undergoing reduction division twice, which is subdivided into meiosis 1 and meiosis 2. So we will see these phases. So when the cells are going to produce the gametes, they replicate the DNA, and this DNA that is replicated will form these homologous chromosomes. The homologous chromosomes have a center that is called the centromere, has arms on top and bottom of the centromeres. That one will be shorter than the other one. The shorter, you will call it the P arm, P for petite, which is small in French, and then the long arm, which is called the Q arm. And then a good student in one of my A&P classes find out why it was called Q. And there is no reason. It's just like if the Q letter follows P. And I'm not going to say who it is, but it is here with us. That student from previous semester. So we were kind of in this situation where, well, why is this, why is that? And I asked, well, why the Q arm is called Q? If P, it stands for petite, which means short, maybe the Q stands for long, and it doesn't. It's just continuation of the letter of the alphabet. But then the Q arm is longer than the P arm. And they both are attached into this centromere, which is the point of attachment of these arms. And then the ends of these chromosomes is the telomere. So then all together, these chromosomes, when they are about to form the gametes, they line up and they form tetrads, groups of 4 sister chromatids. So this is a replicated chromosome from the parental side. This is, in this case, this is the maternal side, this is the paternal side. And then they align, and then they start exchanging information. So then this is what happens in meiosis 1. So you have 2 homologous chromosomes. And then you replicate them. So you form 4 chromosomes that they will attach in the centromere. And then these, during the first phase of meiosis 1, which is prophase, they will exchange genetic information. They will cross part of the arms, and then you will have exchange information from the paternal side and the maternal side. And then ultimately, at the end of meiosis 1, you end up with 2 copies of one homologous chromosome in one cell and 2 copies of one homologous chromosome in the other cell. So that cell is going to be haploid. And the chromosomes are going to be different from the original chromosomes because you have exchanged genetic information. So this is what we call the reduction division. So then from those 2 end cells, you end up with 1 end cell. So these are the phases of meiosis. So you have prophase that can be subdivided into early prophase 1 and middle to late prophase 1. In early prophase 1, since you have already replicated your DNA, you can visibly see these chromosomes. This is what happens even in mitosis. You condense the DNA, and then you have disappearance of the nucleoli, and the nuclear membrane starts to break down. And this is to allow the microtubules that are going to separate these chromosomes to be able to reach. It's like if you go to a place where they fast food restaurant and they give you this hamburger wrapped around. I mean, in a paper. What do you need to do to eat the hamburger? You have to unwrap it, right? So similarly, we have to unwrap the chromosomes so the nuclear membrane starts disappearing during this early phase of prophase. And then in middle to late prophase, your homologous pairs pair up in the synapses to form touching tetrads. And this physical exchange is called crossing over. So that crossing over then allows exchange of material from the DNA from our mom's side and our dad's side. So that's why we end up with traits from both. So this is the only time where we have this physical exchange in prophase 1. And then just as in mitosis, after prophase, your chromosomes align in the center of the cell, the equator of the cell, forming the metaphase plate. So they form a line of chromosomes in metaphase to be ready to be attached in the centromere by the microtubules so that you can split these chromosomes, so that you can separate them. And then in anaphase 1, one of the sister chromatids is pulled from the other sister chromatid so that they go to opposite cells. And this is what is known as independent assortment, because you already have in metaphase 1 a random orientation of these chromosomes, and then you separate them randomly too in anaphase 1. And then just as in telophase of mitosis, telo means ending. So the chromosomes are further separated, closer to what is going to become the daughter cells, and then they will be ready for be subdivided together with the cytoplasm of the cells in cytokinesis 1. And in telophase 1, then you separate these chromosomes. So this is the stages of meiosis 1. Early prophase, visible chromosomes. They start pairing up according to homology, so pair 1 with pair 1, pair 2 with pair 2, etc. And then in mid to late prophase 1, you have physical exchange of chromosomes in crossing over. And then in metaphase, you align them in the middle of the cell, which is the equator, with random orientation. Then in anaphase 1, you start pulling them apart. And notice here how in metaphase and in anaphase, you have part of this purple chromosome into the blue chromosome and vice versa. So that's the physical exchange that happens in prophase 1, the crossing over. And then in telophase, these chromosomes are further apart. Then you make again this nuclear membrane, and then start cytokinesis to divide the cytoplasm. But the sister chromatids are still attached. Now, in meiosis 2, what you do, it is split these sister chromatids so that you can have these sister chromatids going into a cell to form a haploid cell. So in prophase 2, what you do is, if the nuclear envelope reform, you break it down again, and then your sister chromatids remain condensed. Metaphase, aligning the chromosomes in the center, and then start attaching the spindle fibers, the microtubules, into these centromeres. And in anaphase, the microtubules start shortening, so that pulls apart the sister chromatids, and then cytokinesis begins, so you form a pinching of the plasma membrane of the cells, and then in telophase, your chromosomes definitely were separated, and then you have 4 genetically unique daughter cells. And then you form your nuclear membrane. So here are the events of meiosis 2. Prophase 1, you have your sister chromatids attached in the centromeres, visible. you start disintegrating the nuclear membrane if it was reformed, and then metaphase, you form your metaphase plate. Your microtubules start attaching to these kinetochores in the centromeres. Anaphase 2, you start pulling them into opposite poles of the cells and start cytokinesis. And then you, in telophase, form again the nuclear membrane around these sister chromatids that were separated, and then you form a new nuclear membrane. And then at the end of meiosis 2, you have 4 distinct cells with unique chromosomes. So differences between mitosis and meiosis. Well, number of chromosomes. In mitosis, you end up always with the same number of chromosomes as the daughter cell. 46 chromosomes from mother cell, 46 chromosomes the daughter cell. And the information is genetically identical. In meiosis, you start with 46 chromosomes, you end up with 23. So you end up with half of the number of chromosomes. And each daughter cell in meiosis will have different gene combinations. So both of them, similarities begins with DNA replication, and this one goes 2 rounds of phases, from prophase up to telophase. So it will be prophase 1, metaphase 1, anaphase 1, telophase 1. And then going into prophase 2, metaphase 2, anaphase 2, telophase 2. So the purpose of meiosis is to reduce the chromosome number and shuffle the alleles, while in mitosis is to preserve the information. So that's why you shouldn't have technically mutations. So number of cells that you end up in mitosis, 2. In meiosis, 4 daughter cells. At the end of meiosis 2, of course. So any questions about these processes? Okay, now let's move into the anatomy of the male reproductive system. So male reproductive system consists of the gonads, which are the 2 testes, and then the copulatory organ, which is the penis, and accessory organs like the seminal vesicles, bulbourethral glands, prostate gland, that assist in the formation of sperm cells and the maturation of the sperm cells, and then the delivery of the sperm cells to the outside of the body. The sperm cells travel along a long system of ductules, which is called the duct system. And then the accessory glands as a whole secrete a fluid that goes into the ducts that become part of the semen together with the sperm cells. And then external structures of the male reproductive system will be the skin that protects the testes, which is called the scrotum, and the spermatic cord. So here is the mid-sagittal view of the male reproductive system. So here we have the testes located in this pouch-like structure that is called the scrotum. And then posterior to the testes, we have this organ of maturation of the sperm cells, which is the epididymis. It looks like a dolphin. It is basically embracing posteriorly and superiorly and inferiorly the testes. And then here we have a long duct, which is called the ductus deferens, which is located inside the spermatic cord, together with blood vessels and nerves and lymphatics that carries the sperm cells into the ejaculatory duct here in this region. And then here we have the seminal vesicles that secretes a fluid that is rich in fructose that helps to nourish the sperm cells. And then here we have the prostate gland that surrounds this ejaculatory duct, and the part of the urethra that it is called prostatic urethra. So then this is the prostate gland. And then here, near to the base of the penis, it is this gland that is called the bulbourethral gland or Cooper gland. This gland secretes a crystalline clear, thick secretion that clears the urethra, the penile urethra, out of urine, and lubricates the urethra so that the sperm cells can travel fast before ejaculation through this spongy or penile urethra. And then here we have the membranous urethra, which, if you recall, is the shortest part of the urethra, that it is located here in this region of the urogenital diaphragm, the system of muscles that helps to maintain and support these internal organs of the pelvic cavity. And then here's the penis. It has 3 cylindrical structures. We will see more details about this, but this is the spongy urethra that enlarges to form the glands of the penis. And then here we have the cavernous bodies, which are 2 cylindrical structures that has spaces that fills with blood during erection. So we will see better details of each of these organs. So the testicles are pair ovoid structures that measures 4 centimeters to 2.5 centimeters in dimension. They are protected by the scrotum, and in the adult, they are protected by this smooth muscle and connective tissue that is located inside the scrotum. Internally, the testes are subdivided into lobules, and then they are like around 250. Each of them, they have internally a coil ductule that is called the seminiferous tubules, which is where the sperm cells are actually made. And then within these tubules, in their walls, there are these cells that are called the spermatogonia, which are the stem cells that produces the sperm cells. And then in between these cells, we have some cells that are supporting cells that are called the sustentacular cells that produce the testicular fluid that has nutrients that helps to nourish these developing sperm cells. Now, between the seminiferous tubules, there are these interstitial cells that produces testosterone. And then the seminiferous tubules are surrounded by cells that are called myoid cells that contract to help to eject the sperm cells and the testicular fluid out of the testes into the straight tubules that move the sperm into a network of tubules that are near to the epididymis that are called the retotestes. And then from there, the sperm cells will go into the efferent ductules that are going to bring the sperm cells into the epididymis so that the sperm cells can be mature in the epididymis. What nourishes the testes? The blood from the testicular arteries, which are part of the gonadal arteries that arises laterally just below the renal arteries, which are branches of the aorta. And then once the testicles get their blood supply, they will drain that blood supply into the testicular veins. And before that, the blood goes into a network of veins that are interconnected, that are called the pampiniform plexus, that surrounds the spermatic cord. And then the testicles are innervated by nerves coming from the sympathetic and parasympathetic divisions of the autonomic nervous system. And then the testicles are rich in nociceptors, which are receptors for pain, and thermoreceptors. So that explains why the testicles are very sensitive to pain or injury, of course. So here is the view, the mid-sagittal view of the testicles and cross-section of the seminiferous tubules. So if you magnify this region, you can see the testicles, how they are connected into this spermatic cord, which has this vas deferens, pampiniform plexus, and the testicular artery and lymphatics and nerves. The testicular nerves provide sensation to the testicles. So this is a sagittal section, so that what you can see is that these testicles are subdivided into these lobules by this septa that goes from the cortex into the mid portion, which is located in this region. So we have around 250 of these, and inside of these, you can see these tubules that if you make a cross-section of these tubules, you can see the different layers. So this is the most external part of these tubules, which is like the myoid cells. And then inside the myoid cells, you have near to them the spermatogonia, which are the stem cells that are going to produce the sperm cells. And then the spermatogonia will subdivide, forming primary spermatocytes and then secondary spermatocytes. And then these secondary spermatocytes will start forming these sperm cells. So these are developing sperm cells. And then in between these seminiferous tubules, you have the interstitial cells, which are the cells that produces testosterone. And then here you have the sustentacular cells or nerve cells that helps to nourish and protect these different levels of differentiation of the sperm cells. So from external to internal, you have less mature cells to more mature cells. And this will be the lumen of the sperm cells. So this is an actual micrograph of these seminiferous tubules, and showing you that in the lumen, you have sperm cells ready to go into this epididymis to be mature in this region. So then sperm cells are produced here in the seminiferous tubules, then go into these straight tubules, and then goes into these red testes, and then into these efferent ductules, and then into the ductus of the epididymis. And in here, the sperm cells mature, and finally they reach from the epididymis, they start being mature in the head, then the body, then the tail, and then from the tail, they migrate into the ductus deferens, which is also known as vas deferens. So the epididymis, the dimensions, will be 3.8 centimeters in, or 1.5 inches in length. And if you extend the ductus of the epididymis, it will give 19.7 feet, almost 20 feet of length. And just as the seminiferous tubules, which are supercoiled, this ductus of the epididymis is supercoiled, so that allows them to be located in a small space. And then the walls of the epididymis internally has these epithelial cells, which are pseudostratified columnar, that helps to absorb the excess of fluid the testicular fluid and pass nutrients to the sperm to complete the maturation. So the sperm cells can be here stored for several months, if not ejaculated, or and then they can be reabsorbed. Details about the vas deferens or ductus deferens. It's a long, thin tubule. It measures between 40 to 45 centimeters or 15.7 to 17.7 inches in length. And the diameter is very small, 0.1 of an inch or 2.5 millimeters. So once it extends out of the epididymis, it follows a posterior course that embraces the posterior region and superior region of the urinary bladder, and then enters into the pelvic cavity through the inguinal canal to pass over the ureter. And then it enlarges near to the prostate gland into a region that is called the ampulla of the ductus deferens, or ampulla of the vas deferens. And then internally, this vas deferens, it has the same epithelium as the epididymis, pseudostratified columnar epithelium. And then this vas deferens, the next layer outside of this epithelium, is going to be smooth muscle cells that are going to allow the contraction and relaxation so that you will have peristalsis. So you have a longitudinal muscle and a middle circular muscle that helps for the transfer of the sperm to be faster. And as well here, you can have this innervation by the sympathetic system and the parasympathetic system. Now the ejaculatory duct, which is a tiny region, it measures 0.8 inches or 2 centimeters in length, is where the sperm cell enters from the ampulla of the ductus deferens together with fluid from the seminal vesicles and the prostate gland. So it is like a dilated region that helps to mix the contents from the prostate gland, seminal vesicles, and the sperm cells coming from the vas deferens so that you can form semen, which is a homogeneous mixture made out of, well, it's a mixture made out of sperm cells and the fluids of the seminal vesicle and the prostate gland. So this ejaculatory duct empties into the urethra. And then if you remember, the regions of the urethra are prostatic urethra, surrounded by the prostate gland, membranous urethra, surrounded by the external urethral sphincter, and then the spongy urethra, which is located inside the spongy body of the penis, which is the longest part of the urethra in males. And then within this spongy urethra, you have several openings in the wall that allows the emptying of secretions coming from urethral glands that helps as well for the lubrication of the urethra so that sperm cells can travel fast. And then the entire length varies between 18 to 20 centimeters or 7.1 to 7.9 inches. The male copulatory organ is the penis. It has a root which is located nearby to the membranous urethra in the pelvic cavity. And then this will be connected to the body or shaft. And then the body or shaft and in an enlargement at the tip of the penis, which is called the glans penis, that it is protected and surrounded by this skin that is called the prepuce, that will be excised or taken off in males who are circumcised. So adult circumcision can reduce male risk of acquiring infections by 50 to 60%, not only from HIV, but any other infections. And then if you make a cross-section of the penis, you can see that it has 3 cylindrical erectile bodies. Well, the 2 ones that are erectile will be the corpora cavernosa. And then the corpus spongiosum surrounds and protects the urethra. And then the corpora cavernosa has this dense irregular connective tissue and spaces that are filled with blood during erection. And then the corpora cavernosa is split at the base, forming 2 crura, which basically attaches the base to the ischial rami of the coxal bone. And then you have there as well this corpus spongiosum, the starting point of the corpus spongiosum. So the penis is innervated by sensory and motor fibers. And then it has receptors for temperature that are abundant and that are important for sexual stimulation. And then the penis and the scrotum make up the external male genitalia, and this is part of the male perineum, the area between the thighs that is subdivided into the urogenital diaphragm anteriorly and the anal triangle posteriorly. So here is the view of the duct system and the penis from a longitudinal cut view anteriorly. So here are the ureters connecting into a urinary bladder. So this is a urinary bladder. And here is the testis, the epididymis, the ductus deferens. And then this is the ampulla, or the dilation of the ampulla of the ductus deferens. Sorry, this is the closed view. This is the open view. So this is the dilation of the ductus deferens. Why there is this dilation? Well, because ejaculation of the sperm, this area is going to receive the sperm cells, and then it's going to be combining with this fluid from the seminal vesicle. So you have to have kind of a wide area to receive all this sperm volume. So then this ductus ampulla of the ductus of vas deferens, or ductus deferens ampulla, sorry, and these seminal vesicles are going to empty their secretions into the ejaculatory duct. And then the prostate gland is going to have several openings within this region so that it also can secrete its secretions. So over here, that's where sperm cells, prostate gland fluids, seminal vesicle fluid mix to form the semen. And then the semen will transfer into the membranous urethra and then into the penile urethra or spongy urethra. And then here's the base of the penis, or the root. This is the bulb. So here we have the bulbourethral gland or Cooper gland that before ejaculation happens, it releases this crystalline fluid to clear up the urine. And then here's the crus penis that attaches this corpora cavernosa into the ischium. And then here is the spongy urethra, and then this is the corpus spongiosum that surrounds the spongy urethra. This is the glans penis, and then the prepuce and the external urethral orifice. And then this is the cross section of the penis at this region, showing you the dorsum of the penis. So this is the upper part of the penis where there are some dorsal veins and arteries of the penis. And then in the middle of this corpora cavernosa, there are deep arteries. So when blood rushes into the penis, It fills this corpora cavernosa, and that compresses these veins. That prevents temporarily the blood from flowing out, and that creates volume within this corpora cavernosa. That is what it causes erection. Details about the seminal vesicles. They are paired glands on the posterior surface of the urinary bladder. And then they measure 15 centimeters or 5.9 inches in length, but they coil until about 5 centimeters or 2 inches in length. And just as the ductus deferens, they have pseudostratified columnar epithelial cells, and they secrete a fluid, seminal fluid, that is yellowish, that it makes between 60 to 70% of the volume of the semen. So the seminal vesicles then makes more of the semen than the prostate gland. And then again, as I told you before, this fluid is rich in fructose. Fructose is a carbohydrate that it is used by the sperm cells to make ATP. In the neck region of the sperm cells have a lot of mitochondria that produces ATP, so that the sperm cells can move the flagella really rapidly, so that it can swim really fast. And then they have also a component that's called prostaglandins that ensures the contraction of the male smooth muscle cells to propel the sperm cells really fast. So the seminal fluid also has some proteins that are called coagulating proteins. These coagulating proteins, together with an enzyme from the prostate gland, forms a temporary clot of semen in the female reproductive tract that helps for the sperm cells to be able to swim faster towards the cervix and then towards the fallopian tubes. Now, the pH of this fluid in the seminal vesicles is alkaline. That helps to neutralize the acidity of the pH of the female reproductive system. Prostate gland is an egg-sized gland that sits inferior to the neck of the urinary bladder, surrounding the prostatic urethra and the ejaculatory duct. It is made of segments, 20 to 30 tubular glands that are embedded in smooth muscle and dense irregular connective tissue. So just as the seminal vesicles, the prostatic gland has secretions that goes into the ejaculatory duct, and then the fluid is milky in appearance, so it's whitish and makes 20 to 30% of the volume of the semen. The carbohydrate that this prostate gland has is citrate, and it has similar function to the fructose. It helps to produce ATP. And then there is this PSA, or prostate-specific antigen, that is an enzyme that dissolves the clot of the semen. So this is initially formed in the female reproductive tract so that the sperm can move deeper. Now, other components will be antimicrobial chemicals that helps to decrease the risk of infection from the female reproductive system, because there are some bacteria there that helps to acidify the pH. And then the prostate gland also secretes an alkaline fluid that helps to decrease the acidity, to neutralize the acidity of the female reproductive system. Clinical note about the prostate gland, it can enlarge, and this is sometimes benign. This is called BPH, benign prostatic hyperplasia. But sometimes the prostate gland also can develop tumors or cancer. And prostate gland slowly enlarges from birth to puberty to help in the reproductive function. But that enlargement, it's supposed to be up to the age of 30, but if the male has further enlargement after the age of 45, with the number of cells increasing, this is called benign prostatic hyperplasia. Hyperplasia is an increase in cell number, not in cell size. That's not the same as hypertrophy. But that is very common to happen, and that can occlude the prostatic urethra and can cause problems of urination. So the males will have problem urinating because they have an obstruction of this prostatic urethra. Now, risk factors include, for this problem, include genetic predisposition, African ethnic heritage, age over 50, obesity, and poor diet. And after the age of 50, males, they're supposed to have annual digital rectal examination to see if the prostate gland has an enlargement, and as well to measure the levels of PSA. Sometimes the levels of PSA can be suggestive of cancer. Definite diagnosis of cancer will be having a biopsy. So, PSA level below 2.5 nanograms is considered normal, but it can increase as the number of cells increases, and suspicious lumps or PSA levels of greater than 4 nanograms per milliliter warrant for the male to have a biopsy. And then the treatment of prostate cancer can be surgery if it's detected earlier, radiation therapy after chemotherapy and hormone therapy. And then if a male has a slow-growing tumor, it needs to be monitored, and aggressive tumors, they need screening and early detection to prevent fatality. Questions? Okay, so for bulbourethral glands, they are located at the side of the membranous urethra. They have a size of a marble, so they are very small, around 0.4 inches or 1 centimeter in diameter. And then again, secretes a thick alkaline mucus-like secretion that neutralizes the pH of the urethra in males and lubricates the urethra. Semen has 5% of sperm cells, and then the other 95% will be secretions, mixed secretions from the prostate gland and also from the seminal vesicles. So the volume of semen, that is ejaculated is between 2.5 to 5 millimeters, and it's supposed to have between 40 to 750 million sperm cells. And then about 5 minutes after the semen is ejaculated, it will coagulate to prevent sperm cells leaking out of the female reproductive system. But 15 to 30 minutes later, with this PSA, the clot will break down. And then the sperm cells will gradually start going up to the reproductive tract. Once inside the female reproductive system, the sperm cells undergo a process that is known as capacitation. So it helps for the sperm cells to be able to penetrate and fertilize an egg cell. And then you have these prostaglandins that stimulates the contraction of the female reproductive tract together with oxytocin that helps to move the sperm farther into the tract. And then again, you have the release of these antimicrobial chemicals to prevent damaging to these sperm cells. Questions? Okay, details about the scrotum. So the scrotum is a sac that contains the testes, is located in the perineum. It is subdivided into right and left by the septum, the raphe, that is visible externally. And then internally, the scrotum, externally has the most external layer, which is the skin, but internally has the superficial fascia and a muscle that is called dartos muscle. This dartos muscle helps to wrinkle the skin of the scrotum, and that helps to preserve heat during colder weather. Now, during hot weather, the skin smoothens because this dartos muscle relaxes. So this dartos muscle, basically, it is skin muscle. And then the cremaster muscle, which is attached into the vas deferens in the spermatic cord, which is attached into the spermatic cord, it is going down into the scrotum as well, and it helps to pull the testicles up during winter time, and then relaxes this muscle and during hot weather time, so that you regulate the temperature of the testes. Spermatogenesis has to happen at a cooler body temperature than, at a cooler temperature than body temperature. Usually 1 degree Celsius less than body temperature. Because this spermatogonia can undergo spontaneous mutations under higher temperature. Now, the spermatic cord, which is a tube that exits the scrotum, contains the ductus or vas deferens, also contains the cremaster muscle, the testicular artery, testicular vein, lymphatic vessels, and nerves. And this passes through the external inguinal ring and ends at the inguinal canal. And then sometimes, if that canal is wider open, can lead to exit of loops of intestine into what we call the inguinal hernia. So here is the different components of the scrotum and the spermatic cord. So this is the scrotum. This is the fascia. This is the dartos muscle. This is the cremaster muscle. And inside will be the testes. And then here in the spermatic cord, which is surrounded by this muscle. If you remove this muscle, you can see, in part of the wall, you can see these components. So we have the testicular artery, the pampiniform plexus that are going to drain into the testicular vein, which is going to drain into the inferior vena cava. And then you have these nerves and then the lymphatics. And then here we have the external inguinal canal at the end of the inguinal canal. And again, this one can be wide open, and then that leads to protrusion of loops of intestine to the scrotum in what is called the inguinal hernia. So the way to help to control as well the body temperatures for spermatogenesis is through countercurrent heat exchange. So heat from the arterial blood passes into the veins. That's why we have several veins, so that you have more absorptive area, surface area, so that artery transfer the heat into the veins. That helps for the testes to have cooler temperature. And then again, the scrotum can be having the skin wrinkle or not. And then with this contraction of the cremaster muscles, you can pull the testicles up or down so that you can have heat loss. So here is a table showing you functions of the male reproductive system structure. So testes, producing sperm cells, testosterone, and a hormone that is called inhibin. So when the sperm cells are produced, you don't need to produce anymore, so you send a signal back into the hypothalamus and also into the pituitary gland by a hormone that is called inhibin. Females also produce inhibin to help to regulate the production of egg cells. And then the duct system, including the epididymis, transfers sperm cells. And in the case of the epididymis, it specifically promotes sperm cell maturation and storing of sperm cells. Ductus deferens can store also as well sperm cells, but more than likely it's just moving the sperm cells to the ejaculatory duct. Ejaculatory duct transports sperm cells into the ductus deferens, from the ductus deferens, into the urethra, into the ejaculatory place, and then urethra transports the sperm cell out of the penis. Seminal vesicle, secreting alkaline fluid with fructose, prostaglandins, and a coagulating enzyme that helps for the sperm cell to not fall off from the female reproductive tract. And prostaglandin also secretes an alkaline fluid that is rich in nutrients and supports the sperm cell, but in this case, they also secrete the PSA, which is an anticoagulant, and also has immunological functions to prevent damage to the sperm cells by bacteria. Bulbourethral gland secretes this mucus that lubricates the urethra before ejaculation and neutralizing acidity from the urine from the urethra. The scrotum enclosed protects and regulates temperature of the testes. Penis moves urine and semen out of the body and is part of these copulatory organs. Copulation is a fancy word, guys, for sexual intercourse. Questions? Okay, spermatogenesis, the process of forming a sperm cell. So it happens, of course, in the seminiferous tubules, in the testes, and begins in puberty. And a healthy young adult male will make about 400 million sperm cells per day, and it does so by the replication of this spermatogonia, which is the stem cells that are diploid. They have 46 chromosomes. They are found at the most external layer of the ductus deferens. And then once this spermatogonia divides, will form another spermatogonia and a primary spermatocyte. So the one that forms the primary spermatocyte will undergo meiosis 1 to produce 2 haploid secondary spermatocytes. And then these secondary spermatocytes will undergo the second division, which is meiosis 2. And at the end of meiosis 2, you end up with 4 haploid spermatids. Now, The sustentacular cells, which are also known as nurse cells or Sertoli cells, they help to protect the sperm cells by the attack of the immune system because they form the blood-testis barrier. So they connect tightly with tight junctions, preventing the immune system cells to attacking sperm cells. The sperm cells can be attacked because they have a different genetic information from the other cells in the body. So that makes them susceptible to the attack of the immune system. But then with these tentacular cells, you form this blood-brain barrier, blood-testis barrier, and then you protect the sperm cells. And also something that they do is they secrete substances that stimulate this spermatogornia to initiate this meiosis in response to the presence of testosterone and FSH. FSH is follicle stimulating hormone. This is necessary for the sperm cells to be produced, and LH, which is luteinizing hormone, helps for the production of testosterone. So there are several factors then that it is needed for the formation of sperm cells and the protection of sperm cells. And the nurse cells are called nurse cells because they provide nutrients as well for these dividing cells. And now if there will be some damaged cells, these sustentacular cells can phagocytose them, and then they produce a hormone that is called, a protein that is called androgen binding protein that helps to bind this testosterone. And also they produce this inhibin at the end of spermatogenesis. So spermatogenesis is the formation of sperm cells. And then these sperm cells, they need to be shaped so that they can have the definite mature shape of the cells, which is having a diamond-shaped head, and then a neck, and then a long tail. Well, the formation of that shape, it is happening during spermiogenesis, in which the cells change in size and shape. So the spermatids, which are the 4 cells that are produced after the meiosis II has ended, they have kind of this shape, quadrangular or circular shape, and then they start elongating and forming this tail. So that happens during spermiogenesis. And it happens in the epididymis. So this is both spermatogenesis here and spermiogenesis. So you have your spermatogornia undergo mitosis. So it creates another spermatogornia and a primary spermatocyte. This primary spermatocyte enters meiosis I, and it forms 2 secondary spermatocytes that now has 23 chromosomes each, and then at the end of meiosis II, you end up with 4 spermatids, which have 4 spermatids that has these 23 chromosomes each. So these will be haploid cells. And now, these cells that initially were connected, they separate and they were rounded, but they start forming this diamond-shaped head and then this neck with a lot of mitochondria inside and then this long tail. So this is called spermiogenesis, the formation of this mature shape and size. And then this will be spermatogenesis. There is no reason to hear these backpacks packing. Still not the time to leave, guys. I was hearing preparation. Okay, so what happens during spermiogenesis? So in the head, you have the formation of a structure that is called the acrosome. The acrosome is a little organelle that has enzymes that are digestive, that are called acrosins. These acrosins are necessary for the penetration of the egg cell because it will break down a series of cells that surround the egg cells. So between the acrosomes and the nucleus, between the tip of the head and the nucleus, then you have this acrosome, and then the formation of microtubules happens so that the flagella start elongating. So the only human cells that has flagella will be the sperm cells. And then this flagella will measure 55 to 60 micrometers in length. It's a very small unit for an inch. And then it has a very thin width, which is 3 micrometers. And then the mid piece that connects the head to the flagella has abundance of mitochondria so that you can have a lot of ATP so that you can move the tail, which is the flagella. And then it takes around 60 to 70 days for this process to complete. So then this is what happens during spermiogenesis. So you have a circular shaped spermatid or spherical shaped spermatid, and then you start losing cytoplasm, so the cytoplasm becomes smaller, and then all the mitochondria will be clustered into this mid piece, and then here you have the acrosome, and then the nucleus. And then you form the tail, which is the flagella, and then this will be the head. And then the head will devoid of as much cytoplasm, and then you have here the nucleus and the large acrosome, and then the long tail. And then the excess of cytoplasm is shed. And this is a scanning electron micrograph showing you these mature sperm cells. Questions about spermiogenesis? Okay, so one of the main hormones then that regulates this process will be testosterone. And then testosterone is released under the influence of GnRH, which is secreted by the hypothalamus. GnRH stands for gonadotrophin releasing hormone. And then that is released in pulses every 2 hours. And then that GnRH basically is part of a multi-tiered feedback loop that it is called the hypothalamic pituitary gonadal axis. So here are the tiers for controlling testosterone secretion. So in the first tier, GnRH is secreted in a small pulse by the hypothalamus. And this can be in response to feedback from the hypothalamic neurons from a center that is called the arcuate nucleus in the hypothalamus. Then after this, you have the second tier control in which this GnRH stimulates the anterior pituitary gland so that the anterior pituitary gland secretes LH or luteinizing hormone, which is part of what we call the gonadotropins. And then this is also known as interstitial cell stimulating hormone. So that is going to make part of these hormones together with follicle stimulating hormone to go into the bloodstream and reach the testis tissue. So then the third tier control happens when you have LH reaching the interstitial cells in the testicles so that these cells secrete testosterone. And this testosterone now reaches this spermatogonia, and then you start forming this primary spermatocyte, and then this other spermatogonia. And then the FSH will also reach the testicles, but it's going to stimulate the sustentacular cells to secrete androgen binding protein and inhibin so that you don't secrete more of these hormones through long negative feedback inhibition. So hypothalamus secretes GnRH. That reaches the anterior pituitary gland, which secretes FSH and LH. So this is the third tier that goes into the bloodstream and into the testes. So then your sustentacular cells and your interstitial cells secrete testosterone and androgen binding protein and then inhibin so that you can create a feedback loop so that you don't secrete more FSH and LH until you complete the cycle. So that forms the hypothalamic pituitary gonadal axis in males. Exists similarly in females. So something that this ABP does in response to FSH is concentrate testosterone in the seminiferous tubules so that the testosterone won't diffuse, and it helps in the process of making these sperm cells, but also the excess of testosterone will enter into the blood, and that will lead to the development and maintenance of the male secondary sex characteristics, which is having a greater mass density in the bones, the muscles, and also distribution of hair in particular areas, and increase of libido, etc. And then it takes a few years to establish this hypothalamic pituitary gonadal axis, but once it's established, it will remain stable more or less until the age of 70. Any questions? Okay, now about infertility. There are several factors that plays a role in this, but basically, as a definition, is the inability to produce pregnancy after 1 year of unprotected sexual intercourse. And 40% of the cases of infertility happens from the side of the males, which usually will be due to a low sperm count or azoospermia. And that will be a count of less than 50 million sperm cells per milliliter of semen. And that can be due to damage to the testes from either physical trauma, maybe an infection of the testicles, like, for instance, rubella virus infection, radiation, developmental defects, or cryptorchidism, which is when the testicles didn't descend during fetal life. And then the testicles won't be able to produce the sperm cells because they are typically formed initially in the abdominal cavity and they're supposed to descend. But then, if they remain in the abdominal cavity, that high temperature destroys the spermatogonia. So that leads to that. But the most dramatic thing about cryptorchidism is that it can lead to mutations in this spermatogonia and testicular cancer. And then other problems can be lack of secretion of GnRH, so that leads to low levels of FSH and LH, and thus testosterone. So we will leave it up